Alright, in this video I wanted to follow up on one of the phenomenon that we talked about in the video on meiosis, and that is crossing over. So crossing over is one of the specific things that happens during meiosis. It's specific to meiosis. We don't observe it in mitosis. And I wanted to discuss in this video how and why it results in the genetic variation that we see in all of the gametes that we make by meiosis. So let's go ahead and have a look. So first detail that we should always remember is that meiosis has crossing over, and that crossing over occurs during prophase 1 of meiosis. And that is the one and only time that crossing over ever occurs. Again, it does not occur during mitosis, and it does not occur during meiosis 2. So it's only during prophase 1. And we can see that here in this diagram of meiosis. We looked at this in the meiosis video, where we begin with a cell in G1, we go through S phase to duplicate the DNA, go through G2, and then we begin prophase with prophase 1, and then we go through meiosis 1 and enter into meiosis 2. We complete those steps and we create the 4 gametes. Again, it's right here during prophase 1 that we observe the phenomenon of crossing over. Again, the one and only time that it happens is during prophase 1. So let's look a little more closely at the cell in prophase 1 and think about what is going on. So if we zoom in on prophase 1 from that figure, we can again see that crossing over is occurring. And so what's happening during crossing over? There's really two important factors, or two important things going on. So the first is that the homologous chromosomes are paired together. And then, while paired, those homologous chromosomes exchange a little bit of their genetic material. So we can see that because these two homologous chromosomes, the two different color chromosomes, we know they're homologs because they're the same size and shape, but the two chromosomes have exchanged a little bit of their genetic material there and there. That's happened for this other pair of homologous chromosomes as well. The smaller chromosome, the two homologs, have exchanged a little bit of their genetic material at the tip of these chromatids. Okay. So every time crossing over occurs, again in prophase 1, we get pairing of the homologous chromosomes and then swapping of a little bit of genetic material between the two homologous chromosomes. So let's think about those steps and how they work. So why do we get pairing of the homologous chromosomes? Well, that happens because the chromosomes stick together through a process called synapses. So synapses is the pairing of these homologous chromosomes. In this image, one homolog is in red, the other homolog is in blue. Each homolog consists of a pair of sister chromatids. So each of these long structures is one chromatid, but they're held together at the centromere. And again, the two red sister chromatids represent one homolog, or one homologous chromosome. The other homologous chromosome is in blue, and it is also composed of two sister chromatids underneath the red ones here. They're paired up at the centromere as well. So all these little white fibers that are sticking these two homologous chromosomes together are called a synaptonemal complex. They're a bunch of proteins that sort of glue these two chromosomes together during prophase 1 of meiosis. So synapsis causes these two homologous chromosomes to stick together. The next step is that we exchange a little bit of the genetic information. We do that through crossing over. So in this diagram, we're showing two homologous chromosomes, one in red and one in green. The centromere is shown here by this sort of pinched in area near the middle of the chromosome. And again, each homologous chromosome consists of these sister chromatids, the long structures on either side. So there's two red sister chromatids, and there are two green sister chromatids. Okay. Again, synapsis is going to hold these two homologous chromosomes to one another, and their sister chromatids are going to exchange a little genetic information with one another by swapping at some crossing over position. this spot where the two chromatids interact with one another and exchange their information, swap their information, is called a chiasma. And crossing over forms this chiasma structure. So the chiasma is where we swap some of the information, where we transition from the green chromatid to the red chromatid in this chromosome, and from the red chromatid to the green chromatid in this chromosome. So crossing over created these new chromatids that are part green, part red, or part red, part green. Okay. So we took our homologous chromosomes, we paired them up, and we got crossing over. So let's put those steps in the larger view of meiosis. So here is a figure that demonstrates all the steps of meiosis. And again, we're going to begin with a cell in G1, so prior to S phase. And we're going to go through a bunch of steps and generate the 4 genetically distinct haploid gamete cells that meiosis produces. And so along the way, I want to think about what each of these numbers represents. What's going on? So again, we're starting with a cell in G1 during interphase. And at this point, each chromosome consists of a single chromatid. And in number 1 here represents DNA synthesis to create those sister chromatids within each chromosome. So after step 1, we've got the 2 sister chromatids attached to one another at the centromere. That's true for all 4 of the chromosomes that are in the cell. We've got the 2 sister chromatids attached to the centromere, and that's true for the smaller chromosomes as well, 2 sister chromatids attached at the centromere. All right, now that we have our chromosomes duplicated, we will enter prophase of meiosis. So that's number 2. We're going to enter prophase of meiosis, and those homologous chromosomes are going to pair. Again, that happens via synapses, the formation of that synaptonemal complex that holds the 2 homologous chromosomes to one another. So now we've got our chromosomes paired up. In step 3, we're going to get crossing over. So we can see that crossing over has occurred because our homologous chromosome that is mostly red now has a little bit of the other chromosome at the end of this chromatid. And crossing over happens for every single pair of homologous chromosomes in meiosis. So both of our chromosomes have crossed over with one another. We have the large chromosome and the smaller chromosome. Once crossing over has occurred, the cell will continue into metaphase 1, then anaphase 1, and then telophase 1. And so step 4 here represents what takes place during the rest of meiosis 1, where the homologous chromosomes are separated into 2 daughter cells. So we separate the homologous chromosomes during anaphase 1, and after telophase 1, we've got each separate homologous chromosome in its own cell. After meiosis 1, we'll continue on into meiosis 2. In meiosis 2, we're going to separate the sister chromatids that are in each of these chromosomes into their own cells. So at the end, we have 4 individual cells, each containing 1 chromatid, 1 chromosome of each kind, the large chromosome and the small chromosome. And we can see that each of those gamete cells contains a different combination of genetic material. So we can see the very bottom cell has a large chromosome that's all red, but a smaller chromosome that's only part red. The next cell up has a large chromosome that's part red and a small chromosome that's non-red. The next cell up has a large chromosome that's all non-red and a small chromosome that is part red. And then the top cell has a large chromosome that's part red and a small chromosome that is all red. None of these 4 cells contains the same genetic information as any one of the other cells. So crossing over is one of the components that provides the genetic differences that we observe between gamete cells at the end of meiosis. And again, crossing over occurs during prophase 1. because the two homologous chromosomes pair with one another during synapses, and then two of the chromatids, one from each homologous chromosome, swap a little bit of their genetic information. Then when we pull the homologous chromosomes apart during meiosis 1 and the sister chromatids apart during meiosis 2, we generate these 4 cells with genetic differences. So I hope that helps. As always, if you have questions about the process, please let me know. I'll be happy to answer them. And I'll look forward to talking to you again later.